Hey everybody, Dr. Becker. Welcome to chapter 6. This is a great chapter talking about our immune system. So in this chapter, we talk about all of the functions of the immune system, functions of inflammation, hypersensitivity reactions, all sorts of things. This is a 2-part lecture, so this will be part 1. I'm actually going to play the introduction to the immune system from our Elsevier because I think it's a good place to start and launch from. So we're going to watch this here. Here we go.
Despite being surrounded by harmful organisms, toxins, and the threat of our own cells turning into tumor cells, humans manage to survive, thanks largely to our immune system. The immune system is made up of organs, tissues, cells, and molecules that all work together to generate an immune response that protects us from microorganisms, removes toxins, and destroys tumor cells, hopefully, though, not all at once. The immune response can identify a threat, mount an attack, eliminate a pathogen, and develop mechanisms to remember the offender in case you encounter it again, all within 10 days. In some cases, like if the pathogen is particularly stubborn, or if the immune system starts attacking something it shouldn't, like your own tissue, it can last much longer, for months to years, and that leads to chronic inflammation. Your immune system is like the military, with 2 main branches: the innate immune response and the adaptive immune response. The innate immune response includes cells that are nonspecific, meaning that although they distinguish an invader from a human cell, they don't distinguish one invader from another invader. The innate response is also feverishly fast, working within minutes to hours. Get it? Feverishly? That's because it's responsible for causing fevers. The trade-off for that speed is that there's no memory associated with innate responses. In other words, the innate response will respond to the same pathogen in the exact same way, no matter how many times it sees the pathogen. The innate immune response includes things that you might not even think of as being part of the immune system. Things like chemical barriers, like lysozymes in the tears, and a low pH in the stomach, as well as physical barriers, like the epithelium in the skin and gut, and the cilia that line the airways to keep invaders out. In contrast, the adaptive immune response is highly specific for each invader. The cells of the adaptive immune response have receptors that differentiate one pathogen from another by their unique parts, called antigens. Adaptive immunity is also diverse, meaning it can recognize almost an infinite number of specific antigens and mount a specific response against each of them. The trade-off is that the adaptive response relies on cells being primed or activated so they can fully differentiate into the right kind of fighter to kill that pathogen, and that can take a few weeks. But the great advantage of the adaptive immune response is immunologic memory. The cells that are activated in the adaptive immune response undergo clonal expansion, which means that they massively proliferate. And each time the adaptive cells see that same pathogen, they massively proliferate again, resulting in a stronger and faster response each time that pathogen comes around. Once that pathogen is destroyed, most of the clonally expanded cells die off, and that's called clonal deletion. But some of the clonally expanded cells live on as memory cells, and they're ready to expand once more if the pathogen ever resurfaces. Now it's time to meet the soldiers, which are the white blood cells or leukocytes. Hematopoiesis is the process of forming white blood cells, as well as red blood cells and platelets, and it primarily takes place in the bone marrow. Hematopoiesis starts with a multipotent hematopoietic stem cell, which can develop into various cell types. Its future is undecided. Some become myeloid progenitor cells, whereas others become lymphoid progenitor cells. The myeloid progenitor cells develop into myeloid cells, which include neutrophils, eosinophils, basophils, mast cells, dendritic cells, macrophages, and monocytes, all of which are part of the innate immune response and can be found in the blood as well as in the tissues. The neutrophils, eosinophils, and basophils are considered granulocytes. because they contain granules in their cytoplasm, and neutrophils in particular are also referred to as polymorphonuclear cells, or PMNs, because their nuclei contain multiple lobes instead of being round. During an immune response, the bone marrow produces lots of cells, many of which are neutrophils. Neutrophils use a process called phagocytosis. That's where they get near a pathogen and reach around it with their cytoplasm to swallow it whole, so that it ends up in a phagosome. From there, the neutrophils can destroy the pathogen using two methods. They can use their cytoplasmic granules or oxidative burst. First, the cytoplasmic granules fuse with the phagosome to form the phagolysosome. The granules contain molecules that lower the pH of the phagolysosome, making it very acidic, and that kills about 2% of the pathogens. Now, the neutrophil doesn't stop there. It keeps swallowing up more and more pathogens until it's full of pathogens, and at that point, it unleashes the oxidative burst. During an oxidative burst, the neutrophil produces lots of highly reactive oxygen species like hydrogen peroxide. These molecules start to destroy nearby proteins and nucleic acids within the phagolysosomes, which are the components of the pathogen that has been ingested. The net result is that the pathogen is eliminated. Now, in comparison to neutrophils, eosinophils and basophils are far less common. They both contain granules that contain histamine and other pro-inflammatory molecules. Eosinophils stain pink with the dye eosin, which is where they get their name. They are phagocytic cells, even though it's not their primary mechanism of attack. They are best known for fighting large and unwieldy helminthic parasites, or worms, by releasing molecules that can poke holes in the outer layer of helminths. These cells are also involved in allergic reactions, such as atopic dermatitis and allergic rhinitis, also known as hay fever. When involved in allergic reactions, eosinophils degranulate, meaning they release various enzymes and proteins within their granules, and this causes an inflammatory reaction. Next, you have basophils, and they stain blue with the dye hematoxylin. And unlike neutrophils, basophils are non-phagocytic. On the flip side, they have granules that contain histamine and other pro-inflammatory molecules. Therefore, they are important in initiating allergic responses. Finally, there are the mast cells, which live in tissues, not in the blood, and are very similar to basophils. They are also non-phagocytic and are involved in allergic responses. Next up are the monocytes, macrophages, and dendritic cells, which are also phagocytic cells. They gobble up pathogens, present antigens, and release cytokines, which are tiny molecules that attract other immune cells to the area. Monocytes only circulate in the blood. Some monocytes migrate into tissues and differentiate into macrophages, which remain in tissues and aren't found in the blood. Dendritic cells are the prototypical antigen-presenting cell. Dendritic cells are usually found in sites that are in contact with most external antigens, like the skin epithelium or the gastrointestinal mucosa. When dendritic cells are young and immature, they're excellent at phagocytosis, constantly eating large amounts of protein found in the interstitial fluid. But when a dendritic cell phagocytoses a pathogen, it's a life-changing, coming-of-age moment. Mature dendritic cells will destroy the pathogen and break up its protein into short amino acid chains. Dendritic cells will then move through the lymph to the nearest lymph node, and they'll perform an antigen presentation, which is where they present those amino acid chains, which are antigens, to T cells. Antigen presentation is what connects the innate and adaptive immune systems. Antigen presentation is something that can be done by dendritic cells, macrophages, as well as monocytes, which is why all of these cells are referred to as antigen-presenting cells. Dendritic cells are the best at this process because they are the only cells that live where pathogens enter, through the epithelial like skin, gut, and airways. And they are the only cells that can traffic from these tissues to lymph nodes, where T cells circulate. Now, only T cells with a receptor that can bind to the specific shape of the antigen will be activated, and that's called priming. It's similar to how a lock will only snap open when a key with a very specific shape goes in. However, T cells can only see their antigen if it is presented to them on a silver platter, and on a molecular level, that platter is the major histocompatibility complex, or MHC for short. So the antigen-presenting cell will load the antigen on an MHC molecule and display it to T cells. And when the right T cell comes along, it binds. The final group of blood cells, the lymphocytes, includes B cells, T cells, and natural killer cells. B and T cells make up the adaptive immune response, while natural killer cells are part of the innate immune system. B cells and natural killer cells complete their development where they started, in the bone marrow, whereas some lymphoid progenitor cells migrate to the thymus where they develop into T cells. All of the lymphocytes are able to travel in and out of tissue and the bloodstream. Natural killer cells are large lymphocytes with granules, and they target cells infected with intracellular organisms, like viruses, as well as cells that pose a threat, like cancer cells. Natural killer cells kill their target cells by releasing cytotoxic granules. These granules contain molecules that punch holes in the target cell membrane by binding directly to the phospholipids and creating pores, and release some molecules that get inside the cell and cause target cells to undergo apoptosis, which is a type of programmed cell death. B cells, like T cells, also have a receptor on their surface that allows them to only bind to an antigen that has a very specific shape. The main difference is that B cells do not need antigens to be presented to them on an MHC molecule. They can simply bind to an antigen directly. When a B cell binds to a protein antigen that's on the surface of a pathogen, it's capable of internalizing that antigen, degrading it, and presenting it to T cells. So technically, they're also antigen-presenting cells as well. Like other antigen-presenting cells, the B cell loads the antigen onto an MHC molecule called MHC2 and displays it to T cells. When a T cell gets activated, it helps the B cell mature into a plasma cell, and a plasma cell can secrete lots and lots of antibodies. Typically, it takes a few weeks for antibody levels to peak. The antibodies, or immunoglobulins, have the exact same antigen specificity as the B cell they came from. Antibodies are just the B cell receptor in a secreted form, so they can circulate in the plasma, which is the non-cellular part of blood, attaching to pathogens and tagging them for destruction. Because antibodies aren't bound to cells and float freely in the blood, this is considered humoral immunity, a throwback to the term humors, which refers to body fluids. Now, the final type of lymphoid cell is the T cell, and it's in charge of cell-mediated immunity. T cells are antigen-specific, but they cannot secrete their antigen receptor. A naive T cell can be activated or primed to allow it to turn into a mature T cell by any of the antigen-presenting cells, but most often it's done by a dendritic cell. Now, there are two main types of T cells, CD4 T cells and CD8 T cells, where CD stands for cluster of differentiation. There are hundreds of CD markers in the immune system, and these CD markers are useful in telling different cells apart. For example, all T cells are CD3 positive because CD3 is part of the T cell antigen receptor. So CD4 positive T cells are actually CD3 positive, CD4 positive, and these cells are called helper cells because they're like generals on the battlefield. They secrete cytokines that help coordinate the efforts of macrophages and B cells. Helper T cells can only see their antigen if it's presented on an MHC2 molecule. CD8 positive T cells are CD3 positive, CD8 positive, and they're called cytotoxic T cells because they kill target cells, really similarly to how natural killer cells do it, with one major difference. CD8 positive T cells only kill cells that present a specific antigen on an MHC1 molecule, which is structurally similar to the MHC2 molecule, whereas natural killer cells aren't nearly as specific in who they kill. So now let's go through a complete immune response with a bacterial pathogen in the lungs. To start, the bacteria will have to get breathed in, slip by your nose hairs, past the cilia in the airways, and will then have to penetrate past the epithelium layer of the lungs. Once it's in the lung tissue, the bacteria will start to divide and might encounter a resident macrophage in the lung tissue, which will ingest the bacteria and start releasing cytokines. Those cytokines start the inflammatory process by making blood vessels leaky and attracting nearby eosinophils, basophils, and mast cells, which release their own cytokines and granules, amplifying the inflammation. Neutrophils from the blood, as well as fresh new ones from the bone marrow, dive into the tissue and join the battle. If the pathogen was a virus, natural killer cells would help destroy the infected cells at this point. This is all part of the innate immune response. Around this point in the infection, immature dendritic cells residing under the epithelium digest the pathogens and move from the lung tissue over to a nearby lymph node where they present the processed antigen on an MHC2 protein to a naive T cell. The dendritic cell, which is part of the innate immune response, bridges the innate and the adaptive immune responses when it presents the antigen to the T cell, which is part of the adaptive immune response. Sometimes, if the infection is spreading, bacteria might find its way to a lymph node without the help of a dendritic cell. In this case, B cells, part of the adaptive immune response, might directly phagocytose the bacteria and present it to a naive CD4 positive T cell. Either way, if the antigen is the right fit for the T cell will begin to differentiate and undergo clonal expansion. Differentiated CD4 positive T cells will release cytokines that will induce B cells to differentiate into plasma cells, which secrete antibodies that will go into the lymph and then into the bloodstream. The antibodies will tag pathogens, making it easier for the phagocytes to eat them. Some cytokines will activate macrophages to kill bacteria that have been phagocytized, but cannot be killed by the macrophage alone unless it gets help from its friends, the T cells, of course. If the pathogen was a virus living and replicating in the cytoplasm of the infected cells, the CD8 positive T cells would kill any infected cells that express the viral antigen on an MHC1. Over time, as the invading pathogen dies off, most of the B and T cells die of neglect, but a few turn into memory B cells and memory T cells, which linger for years in case they're needed in the future. All right, as a quick recap, the immune system has an innate and an adaptive response. The innate immune response is immediate but nonspecific and lacks memory, whereas the adaptive immune response is highly specific and remembers everything, but it takes several days to get started and almost 2 weeks to peak.
As always, I think Osmosis does a phenomenal job of giving us an illustration to follow along with and breaking it down so it seems a little more simple and straightforward. I love in your book, it starts off with the role of the immune system. The human body is warm, moist, and full of nutrients with a stable pH. All optimal living conditions for many microorganisms. I'm just laughing because I've never really thought of the human body being a warm, moist, and full of nutrients. But the reason that they say that is we are a perfect host for a lot of different things. And so we know that we are under quote unquote attack a lot when we interact with the environment around us. They're also pointing out that there are as many bacterial cells as human cells in our body, and we also could have countless viruses, protozoas, and fungi. Collectively, all of those organisms are known as the human microbiota, and for the most part, they consist of commensal organisms, so things that should be there, that coexist with humans without causing overt disease. Some of them even provide physiological benefits like protection against other pathogens and aiding in digestion and nutrient absorption. However, the human body constantly faces attack by disease-causing microorganisms known as pathogens. The reason we are reviewing this is because we have to remember that to defend ourselves against the pathogens that attack us, we have to be able to maintain our homeostasis. And when we get away from homeostasis, then we have problems. So working in synergy with commensal organisms, we as humans have evolved a really highly complex and highly orchestrated system of cells, tissues, and organs that are collectively referred to as our immune system. Immune cells produce the numerous molecules that support the antimicrobial functions, cell-to-cell communication, and cell growth, differentiation, and movement, right? In addition to external threats, the immune system is capable of responding to abnormal self-threats, such as the mutated self-proteins that are expressed by tumor cells, for example, or organs received from a donor in case of a transplant. Because of this, the immune system is a multifaceted defense force that serves to protect the human body from infections, toxins, cancers, and any tissue that is detected as an invader or non-self. The cells of the immune system then are circulating in our bloodstreams to the body tissues via the lymphatic system and back to the bloodstream. They're conducting constant surveillance and attacking invaders. Like any other body system, though, when the immune system fails to function appropriately, then we have problems. This can occur from inherited or spontaneous mutations in critical genes encoding molecules of the immune system, or it can be acquired from infectious or environmental exposures. Failures of the immune system to function appropriately can result in an aberrant immune system response to harmless molecules in the environment, right? So that could be like our allergies, or to self molecules, which is how we end up with autoimmune diseases. Alternatively, the immune system might fail or weakened, and then it doesn't respond to pathogens, and it fails to maintain homeostasis with our harmless microbes, then which can lead to immunodeficiency. So the first concepts that they want us to review here are that some of these terms. Anything can cause edema, anything can cause inflammation. These are general terms. When we talk about hypersensitivity and atopy and allergies, think of things like hives, eczema, allergic rhinitis. Then autoimmune diseases are the things like type 2 diabetes, rheumatoid arthritis, etc. So we talk about the 3 lines of the human defense. Again, I've linked that first osmosis introduction video here if you need that link. So our first lines of defense. So this graphic here is showing us that out in the environment, we have all sorts of pathogens, our viruses, our bacteria, etc. Then we have physical and chemical barriers, such as our physical barrier of our skin. And then within that, we have innate immunity and our adaptive immunity. We know that the skin is our primary source of defense. It's the first layer, then, the primary source of defense. The skin covers the outside of the human body with an outer layer of dead cells and then underlying layers of densely packed living cells, which is our epidermis and our dermis. And those layers provide a strong physical barrier against penetration of pathogens. We regularly shed the outer dead cells of the epidermal layer, and that's a way we also physically shed microbes. So here, this upper top part here is our epidermis, and then our dermis. We can see this top here is where the dead skin cells come to. Okay. So let's review the skin and these defenses. So the epidermis is our main outside layer here. This is where we're going to shed our dead cells and also shed layers of microorganisms. When we get down, more down into the dermis, this is where we keep our sweat ducts and our sweat glands. The skin then, because of that, produces chemical defenses against invasion and colonization. So when we think of the skin as a defense, we think of a physical barrier, but we also have to remember it's a chemical barrier because we secrete sebum. And the sebum is a low pH, it's oily and it's a waxy substance. And then we also produce our sweat, which is high in salt, and that has antimicrobial enzymes. And those are secreted by the sebaceous glands and the sweat glands, and these exist within the dermis. The antimicrobial peptides that are secreted by the skin, such as cathelicidins, can never say that word, and those can be directly antimicrobial. So that's awesome. Our sweat can be very protective, and also those cathelicidins can also activate the production of other effector immune molecules. When we have that acidic, salty environment of our sweat, it also is a production of antimicrobial peptides and enzymes, which then all function together to limit or prevent microbial growth on the skin. So sweating is good for us. When we think of skin, we also think of mucosal surfaces that line the cavities of the body that are exposed to air and ingested substances. So when we think about our mouth, even though we feel like the mouth may be on the inside, it's also part of our skin defense. Think of the mucosa down into our throat and into our lungs. In contrast to skin, which is that outer layer, right? The outer layer of the mucosa is alive and contains specialized cells that produce mucus, which is that thick, viscous fluid that coats the outer layer of cells and functions then to trap and expel microbes. The cells that line the respiratory tract have those short hair-like projections on the surface, which we know are called cilia. When you are in healthy state, the cilia beat in tandem to expel particles trapped in the mucus up and out of the upper and lower respiratory tract. Failure of the mucociliary transport system then is what often leads to lung disease. Think pneumonia, COPD, those things. Cigarette smoking and certain e-cigarettes, now we know, will impair the structure and function of the cilia. So the mucociliary system becomes broken and fails, and that's what contributes to the development of smoking-induced respiratory disease like COPD. The antimicrobial enzyme lysosome is abundant in our mucus and in our tears, and they act as chemical barriers for our mucosa surface. So our tears are functioning to protect our eyes and that layer that's exposed to the air. Lysosome functions by disrupting the cells and the cell walls of certain bacteria, causing that bacteria then to rupture and die. The GI system has its own immune system, and we just touch on it briefly in this chapter, but we will really get into that in chapter 13, which is the GI tract. This one here is what we're talking about in terms of the respiratory mucosa here, then those cilia, and the columnar epithelial cells are here, and these are those goblet cells that we'll talk more about, especially when we talk about the lungs further. We have our nucleus here, a basal layer, a basement membrane, the lamina propria, and our blood vessels here. These are the regions that will physically interact with substances and microorganisms in the environment, but they also will interact with the commensal organisms that are good. Okay. It's really important that you understand the concept of innate versus adaptive immunity. I think that the Elsevier did a great job telling us about this and what the differences are. Really important. That's also what is, sorry, slide, what was talked about in that video. So our innate, so when we say innate, what's natural within ourselves, what is a natural response that we don't even really have to think about, and it's rapid, but it's non-specific, right? So when my innate immune system wants to just fire off a fever, that can happen to 1,000 things can give me a fever, right? It's non-specific. It's just the reaction immunity that we have when our body says, uh-oh, boom. It's kind of like a kid that acts without thinking first. It just throws everything out there in panic. In my head, I'm a very visual learner and I like analogies. So in my mind, our innate immune system is like a guard tower in our compound. And so we're in the center of the compound and we have out past our fences is the wilderness, right? And we have all of those sensors out there. And so our innate immune system is when all of a sudden we get a sensor beeping in the control tower that something has triggered the alarm. We have no idea what it is, but somebody just hits the red panic button. And all of a sudden below us, when we look down, all of a sudden you just see this flood of the soldiers that are just running out with guns and rushing towards all parts of the fence, because that's just reactive. It's a rapid response. And we send out these things that are rapidly produced. So these soldiers that go out we don't really care about because we can make millions of copies of these no problem. So we just send them out there, even though we don't even know what we're fighting against yet. So that's our innate immune system. Something triggers the wires, an alarm goes off. We don't even know what it is, but automatically our body hits the panic button and we just flood the area with our defense system of these immediate things that have a short lifespan, but we can make millions of them. It's a broad response, and it's orchestrated by the circulating white blood cells and tissue-resident scavenger cells. So if you think about that, we know we make our white blood cells and they're going, but also scattered throughout the tissues. So out by the fences, we have other little scavengers that are kind of also listening. So maybe something doesn't actually trip the wire, but they hear something on the other side of the fence and they're going to go explore and be there and be ready. So the tissue resident scavenger cells and the circulating white blood cells will quickly recruit additional immune cells to sites of infection and trauma using chemical gradients and signals from molecules known as cytokines and chemokines, okay, which is like what we have in here. So to me now, the alarm's been triggered, all of these white blood cells go rushing out, the scavenger cells that were already posted near the fences come together, and they quickly are like, yep, we have a problem, and they start sending up the flares when they identify what part of the fence has been breached. And they send up those chemokines and cytokines signaling the problem's over here, the problem's over here. That triggers and turns everybody on. And in my brain now, they're like little Pac-Man men, and they come rushing over gobbling, and they just start ingesting and destroying any evading microorganisms. They don't care what it is. They're not specific. They're not selective. They just kill. The cells of the innate immune system are generated continuously from our bone marrow stem cells. The book refers to it as our multipotent bone marrow. We just pump these bad boys out hour after hour after hour for the duration of our life that comes from our stem cells. And they can rapidly proliferate. They're created very quickly in response to appropriate signals of infection and pathogen invasion. So it's like once somewhere in a different control tower in our bone marrow, once they hear that the alarm's been triggered, man, they just boom, they hit their button and the floodgates open, and they're very quickly able to throw out the rest of the immune cells that we need, our white cells, etc. Molecules of our innate immunity also include, proteins of the complement cascade. So now, we're out in the yard with the fence. We have all of our cytokines and chemokines, all those soldiers, all the white cells and our scavenger cells have sent out the flares of the chemokines and cytokines. Everybody's alerted to the area that we all need to rush to. But also we have other little things at different guard points throughout the yard and the fenced-in area. And those are the proteins of the complement cascade. And the complement system is a system that will also directly attack and lyse bacteria while promoting other aspects of innate and adaptive immunity. So the timing of the cell and molecular innate responses is crucial to contain the replication of pathogens and to prevent the spread of the disease in the host. Okay, so all of this kind of comes together. And this figure here is showing us that we have the cellular and molecular communication between the innate and adaptive immune cells. Both arms of the immune system have signals that modulate the response of the other. So the antigen here, co-stimulation, the chemokines and cytokines, the antibodies, etc. Innate immunity is generally very efficient. It works pretty well for us, and it's not specific to any particular pathogen. So again, it doesn't matter if what's attacking the fence and our perimeter is a virus, if it's a protozoa, if it's bacteria. Our body doesn't know that at the time, so it just, the response is the same. It doesn't matter if it's Staphylococcus, if it's Pseudomonas, it doesn't matter. Our body doesn't care. It's rapid, it's fast, and it's non-specific. However, when we think about the adaptive immune system, it's kind of a different story, okay? I'm going to break here for a minute that we just quickly reviewed again the innate system. Also, our first video talked about it. And let's jump to the next video that's more specific on the innate immune system.
Your immune system is like the military, with two main branches, the innate immune response and the adaptive immune response. Key features of the innate immune response are that the cells are nonspecific, meaning that they don't distinguish one invader from another invader. Also, the response is really fast, happening within minutes to hours, and there's no memory associated with the innate response. In other words, the innate response will respond to the same pathogen in the exact same way, no matter how many times it sees the pathogen. The innate immune response includes things that you might not even think of as being part of the immune system. Things like chemical barriers, like lysozymes in the tears and a low pH in the stomach, as well as physical barriers, like the epithelium in the skin and gut, and the cilia which line the airways to keep invaders out. Now, if a pathogen happens to get in, then the immune system kicks in, and it usually starts with a macrophage, which is the garbage truck of the body. Macrophages eat up dead and dying cells, so that the tissue doesn't get cluttered with them, and that makes room for new cells. They also eat invading pathogens. Since macrophages live in the tissue, they start recognizing pathogens within minutes of an infection. And the way that a macrophage figures out if something is a healthy host cell or a pathogen is by the molecules that a cell or pathogen has on its surface. This is because cells of the innate immune response don't distinguish one invader from another invader. This probably won't come as a surprise, but pathogens have molecules that humans don't have, and they're called pathogen-associated molecular patterns, or PAMPs. PAMPs include bacterial wall components like peptidoglycan, lipopolysaccharide or LPS, and lipoteichoic acid. PAMPs also include fungal wall components like mannan and flagella proteins, which can be found on some parasites and bacteria. For intracellular pathogens like viruses, PAMPs might include the viral RNA or DNA. Now, PAMPs are recognized by pattern recognition receptors, or PRRs, which are receptors on various immune cells including macrophages, neutrophils, eosinophils, basophils, and mast cells. There are two main groups of PRRs, phagocytic PRRs and signaling PRRs. Phagocytic PRRs bind to PAMPs so that a phagocyte can gobble it up, but they do not allow for cytokines to be released to other cells. And this is a super important point, because it allows the macrophage to investigate and eliminate the threat before signaling that there's an invader. This is kind of like investigating a noise before calling the cops.
Stop the video there, and we're going to come back to that in a little bit, but I want to kind of keep with the flow of the text and of text. That's a lot to say. So we'll come back to that here in a moment. When we talked about the overview of the adaptive immune system, a couple things to remember. It's a slower response than the innate immune system. And the adaptive immune system, though, is highly specific for particular pathogens, and they will quantitatively and qualitatively improve during the course of the response. So as they get better with time and with practice. Upon resolution of the immune system response, some small number of these highly specialized cells are retained in the body, and they can actually persist for decades. Some of these cells are things like the T cells, the T helper lymphocytes, for the example in the book, are modulatory and function to promote the activity of other cells. Then we have the other adaptive immune cells, the B lymphocytes, the cytotoxic T lymphocytes, or the CTLs. And those are effector cells that are directly involved in targeting and destroying antigens and foreign cells. So then upon any future encounter, these effector memory cells are able to efficiently promote pathogen neutralization and elimination very quickly, so that you may not even know you were ever infected. You don't have any signs of infection. So that's a really cool feature of the adaptive immune system, because it's known as immunological memory. Immunological memory by the adaptive immune cells is the mechanism upon which vaccinations are based. So if you ever were to think about how does a vaccination work, we'd say through immunological memory. The B cells and the T cells are part of the adaptive immune system. Both the B and T cells recognize highly specific parts of the pathogens called the antigens. So here's an important distinction, and I've learned this 1,000 times, and I swear I forget it every time. But the important distinction here is what the difference is between the B cells and the T cells. And that video of Elsevier did a great job explaining it. The B cells recognize free antigens located in the extracellular compartments of the body. But the T cells can recognize both intracellular and extracellular peptide antigens. But the T cells require other cells to present to them the peptides on that cell surface molecule called the MHC, the major histocompatibility complex. And the Elsevier osmosis did a great job of saying it has to be served up on a silver platter on these complexes. So the complexes are either the MHC or the HLA complex, which is the human leukocyte antigen complex. So the B cells can recognize any free antigen. They're kind of free-loving people out in that extracellular compartment of the body. But the T cells are the hoity-toities that are, their noses up and they're like, no, I need to have something served to me on that silver platter. So if you want me to recognize your antigen in both your intracellular and extracellular compartments, I need that those to be presented to me on the MHC or the HLA complex. A given B or T cell always only recognizes one type of antigen. So each B and T cell has a single specificity. So we're very different now in the adaptive immune system than in the innate. When activated, the B cells synthesize and secrete highly specific Y-shaped molecules called antibodies that can inactivate, neutralize, and label pathogens and toxins. B cells express the same antibody on their cell surface, which forms that antigen recognition component of the B cell receptor, which is the BCR. Signaling a naive or antigen inexperienced B cell through the B cell receptor, the BCR, that is what enables that B cell to become activated and produce antibodies. Okay, so the B cell has to recognize a specific part of the pathogen, which we call the antigen. The B cells will recognize free antigens in extracellular components of the body. It will only recognize one type of antigen, so it has a single specificity. When the B cell is activated, the cells synthesize and then secrete an antibody, which is a specific Y-shaped molecule that can inactivate, neutralize, or label the toxins. The B cells express the same antibody on their cell surface, which then forms the antigen recognition component of the B cell receptor, which we call BCR. When you have a naive or antigen inexperienced B cell, through that BCR, it is then enabled that B cell to become activated and make those antibodies, which are those highly specific Y-shaped molecules. Activated B cells will start to clone and divide, and that is what we call clonal expansion. It's through the clonal expansion that many thousands of B cells can result. And those thousands of B cells all recognize and produce antibody for the same antigen. The antibody-mediated immune response is sometimes then referred to as the humoral immune response. Okay, so T cells. T cells are also part of our adaptive immune system. They are also going to recognize antigens, but the T cells can recognize them intracellularly and extracellularly. But the T cells require other cells to present the peptides, the antigens, to them on that silver platter. And the two types of the silver platter, so to speak, is the MHC or the HLA complexes. The T cell will recognize antigens through highly specific receptors known as their TCRs, T cell receptors, and those are also on their cell surface. The TCRs are present on the surface of each individual T cell, and there's many copies, right? So there's a whole bunch of the same TCRs on the cell surface. The TCRs of the cells, though, each individually are the same specificity. And the T cells have a large genetic variation that is introduced through the somatic gene recombination events in our thymus. So there are millions of different T cells that are present in the body, but each of them have their own TCR specificity and the capacity to proliferate upon recognition of their specific antigen. So the T cells are a little different and they're a little more hoity-toity. There are two types of T cells. So we have the CD4, which is the helper cell. And then we have the CD8, which is referred to as the CTL. Sometimes when you're reading other texts or other materials, sometimes they're just called the helper cells or the killer cells or what have you. So some of these things have some different names, and that can be a little confusing. The CD8 T cells function to lyse the cells infected by intracellular bacteria, virus-infected cells, or tumor cells. CD refers to the cluster of differentiation and signifies specific membrane proteins found on the surface of lymphocytes and many immune cells. In your book, Table 6.1 shows these characteristics of the innate and adaptive immunity, and this is when we start talking about the PAMPs and the DAMPs and XYZ. So as we move forward, we're going to talk more about how the innate and adaptive immune systems work together, and they can be interconnected and oftentimes actually overlap. And we'll kind of jump more into some of these processes. So the CD4 cells are the helper cells, the CD8 cells are the direct killing cells. The book moves on to the functional anatomy of our immune system. So we have specialized organs and tissues that are termed lymphoid organs and lymphoid tissues. If you go to figure 6.4 in your book is where we're at here. The primary lymphoid organs and tissues are the site of the immune cell development, and those include the thymus, which is where the T cells develop, and our where B cells develop. So when we're talking about similarities and differences between B cells and T cells, another difference is that B cells come from the bone marrow, which is why they're the B cells, and the T cells come from the thymus, which is why they're the T cells. Both the T and the B cells develop from our precursor stem cells that reside in our bone marrow. The secondary lymphoid organs and tissues are where they're the sites of the adaptive immune system cell activation, and those are lymph nodes, lymphatic vessels. And in those is where the cells respond to damage and infection of the tissues and the spleen, where immune responses to blood-borne pathogens are initiated. So the lymphatic network provides the T and B cells with the ability to circulate then through the tissues, our lymph system, and our blood system, allowing them to constantly patrol and survey the body for signs of infection. So when we're talking about when we're talking about our innate immune system, our cytokines and chemokines are out there and get released and work with our white blood cells, and they're very fast and non-specific, and they're kind of, they just follow a pre-programmed script. They don't think on their own at all. Scattered amongst the yard with the fence, so scattered out there, we also have these B cell and T cells that are specialized soldiers. They're very smart. They're very bright. They're really good at their job. They're just going to thumb their nose up at those neutrophils and whatever, because they're like, you silly people, you're so, beneath me. I'm a B cell and I'm a T cell. I have special functions and special jobs. So I'm always out there circulating around and patrolling for any signs of infection as well. For those of us that don't know, that's where our thymus is located. Otherwise, this is the nice image of the immune system anatomy. You can see here we have the Peyer's patches, is something you may not have heard of in the past, and that's where they lies within the intestinal wall. Remember that you have intestinal lymph nodes, we have axillary lymph nodes, right? All those little green peas are lymph nodes that we have everywhere. And so when you're doing a good physical exam and assessment, there's specific areas that we teach people to look at and to palpate to feel for the different sites of lymph nodes. All right. Jumping into hematopoiesis. So hematopoiesis in humans occurs in our bone marrow. Remember, we keep saying this is where that multipotent precursor stem cells come from, okay? So we call those hematopoietic stem cells. So our precursor stem cells, the hematopoietic stem cells, are supported to survive, proliferate, copy themselves, and differentiate by the mesentery stem cells, I cannot say this word either, mesenchymal stem cells. So the hematopoietic stem cells are self-renewing, so they can divide to make more precursor cells, and they can develop into the major cellular constituents of blood. So these hematopoietic stem cells, which they refer to as HSCs, those are the ones that can develop into red cells, platelets, and white blood cells. So they're part of the innate and adaptive immune system. So the three bloodlines that come from our precursor stem cells, which are our hematopoietic stem cells. So our three bloodlines come from our HSCs, and we have, those three major lineages, right? So white blood cells, red blood cells, and platelets. So the erythroid line develops into red cells. So from the erythroid line, we get erythrocytes, and the platelet-producing megakaryocytes, and the myeloid lymphoid line, which develops into our white blood cells or leukocytes and myelocytes. That myeloid line originates from a specialized progenitor cell derived from our hematopoietic stem cells, known as the common myeloid progenitor. The common myeloid progenitor can differentiate then into granulocytes, which are our neutrophils, eosinophils, basophils, and mast cells, as well as our mononuclear cells, which are our monocytes, macrophages, and dendritic cells. So if I were taking notes right now, this is a great example and illustration here of the things we're talking about, right? So remember that they can self-renew. So we have our multipotent precursor stem cell here, our hematopoietic stem cell, HCS, and they are going to differentiate and proliferate. Okay, so they split and recreate themselves. And then one of them is going to be able to come back to continue to stimulate more stem cells production. The remaining cells go down one of the two pathways depending on the signal it's received. So it's either going to go down the lymphoid pathway or the myeloid pathway. If it goes down the lymphoid pathway, then you could get a lymphoid stem cell. If it goes down the myeloid pathway, you get a myeloid stem cell. Okay? So we have different types of our stem cells. Okay, so then we can take that into the next step further. So right here is where we have this myeloid stem cell. So remember, we started off with this very early hematopoietic stem cell, the HSC. It split. We're going to follow this myeloid pathway now. Okay, so we're in the myeloid pathway. So we have granulocytes, and under the granulocytes are mast cells, basophils, neutrophils, and eosinophils. And then agranulocytes are our monocytes, the macrophages and the dendritic cells. So then we can divide out this myeloid stem cell is going to go down one of these ways. Okay. If it goes to be a megakaryoblast, which becomes a megakaryocyte, which becomes our platelets. Then we said they could also become a mast cell. It could then also go to be a proerythroblast, reticulocyte, erythrocyte. Or it can become a myeloblast, which then will differentiate out into basophils, neutrophils, eosinophils. And then it also could be a monoblast, which becomes a monocyte. And then the monocyte is what becomes a macrophage and a dendritic cell. Okay. Are you going to remember this for the rest of your career as an APRN? Maybe not, depending on what line you go into of practice. But this is important to understand, especially in school and as you're moving forward. So when we go back to this slide here, right, this all stems from the basic multipotent hematopoietic stem cell, and we're going down the myeloid progenitor line here. Okay, so let's go to the other side. So instead of going this way, now we're down this way. So we're on the common lymphoid progenitor stem cell. And that then becomes into a lymphoblast. And the lymphoblast will become either a B cell, a T cell, or a natural killer cell, which is known as the large granular lymphocyte or innate lymphoid cell. Or the lymphoblast can become also a dendritic cell. So if you wanted to know how does a dendritic cell form? The answer is it could come from either a myeloid stem cell pathway, after a monocyte can then become a dendritic cell and a macrophage, or it can come down the lymphoid progenitor side, in which point the lymphoid cell becomes a lymphoblast, and the lymphoblast then can create a dendritic cell. So we got a couple of different pathways going on here. It's talking about here in this initial differentiation in the lymphoblast, we have to know, is it going to be a T cell or a B cell or an innate lymphoid cell, which includes our natural killer cells and the innate lymphoid cells. And then the further maturation of the T lymphocytes will occur in the thymus. So this is a lot of talking. I know we're an hour into the lecture, but I want to stop and actually play again for you this one here, which is the cell-mediated immunity. And let's listen. It's a good recap.
The key cells of the adaptive immune response are the lymphocytes, the B and T cells. And there are two types of T cells: T helper cells, which express CD4 on their surface, and cytotoxic T cells, which express CD8 on their surface. Helper T cells primarily support other immune cells, whereas cytotoxic T cells kill cells that are infected with a pathogen or are cancerous. Cell-mediated immunity refers to the part of the immune response that's based on cellular interactions, and cannot be transferred through serum from one person to another. That makes sense since CD4 helper T cells interact with other immune cells to stimulate them. Now, when a T cell is initially formed, it's considered naive. This naive T cell is a bit like a student in school that isn't ready to choose a career path. Later, when that T cell encounters an antigen, it gets activated, or primed, and turns into an effector T cell. This process of priming requires two signals. The first signal is the antigen itself, which is usually presented on an MHC molecule on the surface of an antigen-presenting cell like a macrophage or dendritic cell. This antigen has to bind perfectly to the T cell receptor. The second signal is called co-stimulation, and that's when a ligand called CD28 on the surface of a T cell binds to a ligand called B7 on the antigen-presenting cell. This region, which includes the T cell receptor, which binds to the MHC antigen, and CD4 and CD28, which bind with B7, is called the immune synapse. Once the T cell receives both of these signals, a number of changes occur within the cell that transforms the naive T cell into an activated T cell. An activated T cell is one that's ready to become an effector T cell, and the developmental path it chooses often depends on the cytokines in the environment. Going back to the student analogy, an activated student is one that's gotten the signals it needs to graduate from college and is finally prepared to choose a career path. Now, within the broad category of T helper cells, there are subcategories like T helper type 1, or Th1, T helper type 2, or Th2, T helper type 17, or Th17, and T follicular helper cells, or TFH. There's also a class of T cells called T regulatory cells, or Tregs, but these aren't considered T helper cells because they're mostly involved in contraction of the immune response. Within the immune synapse, antigen-presenting cells, or APCs, send out different combinations of cytokines, depending on the type of pathogen. These cytokines help tailor the immune response to the specific pathogen. The activated T helper cell also begins making lots of cytokine IL-2, and it upregulates its IL-2 alpha receptor. The IL-2 receptor has three protein components, alpha, beta, and gamma. A naive T cell only expresses the beta and gamma components of the IL-2 receptor, but these have low affinity for IL-2, in contrast to the alpha component, which has a high affinity for IL-2. Using just the beta and gamma components is kind of like trying to eat an apple with only your lips and tongue, and upregulating the alpha component is like using your teeth to take a big bite out of that juicy apple, much more effective and satisfying. As a result, active T cells bind to IL-2 that they make, a form of autocrine stimulation, that is to say, a cell stimulating itself. In response to the IL-2, the activated T cell starts to rapidly undergo cell division, a process called clonal expansion. Now, let's go through the types of T helper cells, the career paths, one at a time. Macrophages and dendritic cells infected with viruses and intracellular bacteria produce IL-12, and natural killer cells produce interferon gamma. In addition, any virally infected cell will produce interferon alpha and interferon beta. When these cytokines are found in the same area as a T cell that's becoming activated, they bind to the T cell and cause it to make transcription factors like STAT1, STAT4, and TBET. These transcription factors cause a number of genes to get expressed within the T cell, transforming it into a Th1 cell. A Th1 cell then secretes interferon gamma, which reinforces the process. Interferon gamma also boosts the macrophage's ability to kill ingested pathogens by increasing their production of reactive oxygen species, nitric oxide, and lysosomal proteases. Th1 cells also express CD40 ligand on their cell surface, and that binds to the CD40 receptor on macrophages, which also stimulates them. Th1 cells also produce IL-2, which makes natural killer cells, CD8-positive T cells, and B cells proliferate. So in short, Th1 cells stimulate themselves, and they stimulate a number of other immune cells as well. When Th1 responses are not carefully controlled, this can sometimes lead to autoimmune reactions. Next up are the Th2 cells. When there's a parasitic worm that's too large to get phagocytosed, it gets destroyed by eosinophils, basophils, and mast cells, which dump their toxic granules full of proteases and hydrolases. This kills the parasite, and its antigens get picked up by scavenging macrophages and dendritic cells. These antigen-presenting cells produce IL-4, IL-5, and IL-10 within the immune synapse with a T cell. When these cytokines are released with an immune synapse, they bind to the T cell and cause it to make transcription factors like STAT6 and GATA3. These transcription factors cause a number of genes to get expressed within the T cell, transforming it into a Th2 cell. Once it's become a Th2 cell, it produces cytokines like IL-4, IL-5, IL-10, and IL-13, which stimulate B cells to make IgE, which can coat the worms. In addition, Th2 cells stimulate mast cells and basophils to bind the Fc region of IgE and degranulate, so that they can kill the parasite. Additionally, IL-5 plays a key role in recruiting eosinophils to the site of infection, where they accumulate and degranulate in order to kill the invader. Unfortunately, Th2 responses can sometimes get directed at non-pathogenic antigens, like peanuts or tree pollen, and this results in allergies, atopic dermatitis, and asthma. IL-13 specifically stimulates mucus secretion and intestinal peristalsis, which can help remove parasites from the intestines. Finally, the IL-4 and IL-5 help to stimulate the Th2 cells themselves, which reinforces the process. Now let's move on to the Th17 cells. Extracellular bacteria and fungi are often picked up by dendritic cells or macrophages, which recognize bacterial peptidoglycans, lipopeptides, or fungal glycans. These antigen-presenting cells produce IL-1, IL-6, IL-23, and TGF-beta within the immune synapse with a T cell. When these cytokines are released within an immune synapse, they bind to the T cell and cause it to make transcription factors like ROR-gamma-T and STAT3. These transcription factors cause a number of genes to get expressed within the T cell, transforming it into a Th17 cell. Once it's become a Th17 cell, it produces cytokines like IL-17, which is where it gets its name. The main function of IL-17 is to recruit neutrophils, which engulf these extracellular pathogens and destroy them. When Th17 responses aren't carefully controlled, they can lead to autoimmune disease and asthma. Finally, there are T follicular helper cells, which are a special group of T cells that live in the follicles of lymph nodes. Their primary role is to help generate memory B cells. Occasionally, antigen-presenting cells produce IL-6, IL-21, and IL-27 within the immune synapse with a T cell. When these cytokines are released within an immune synapse, they bind to the T cell and cause it to make transcription factors like BCL6 and CMAF. These transcription factors cause a number of genes to get expressed within the T cell, transforming it into a TFH cell. Once it's become a TFH cell, it expresses CD40 ligand on its cell surface, and that binds to the CD40 receptor on B cells and makes them express cytokine receptors. The TFH cells also produce cytokines like IL-21 and interferon gamma, which stimulate B cells to differentiate into memory cells, or plasma cells that primarily produce IgG antibodies. Alright, as a quick recap. Immune responses are determined by the cytokines the T cell is exposed to within the immune synapse at the time of activation. Th1 cells fight intracellular infections. Th2 cells fight parasites. Th17 cells fight fungal and bacterial infections, and T follicular helper cells help establish memory B cells.
Jump into this video here.
Lymph means clear water in Latin, and it describes the fluid that flows through the lymphatic vessels and lymph nodes, which make up the lymphatic system. The three major roles of the lymphatic system, the reason we need it in the first place, are that it returns fluid from the tissues back to the heart, it helps large molecules like hormones and lipids enter the blood, and it helps with immune surveillance to keep infections from running amok. So let's take a closer look at lymph and where it comes from. The blood in the arteries is under a lot of pressure because it needs to reach every little nook and cranny of the body. Those arteries branch out into narrower and narrower arteries, and then arterioles until finally it gets to the capillaries, which have walls that are only 1 cell thick and are slightly porous. Red blood cells are too big to fit through the capillary pores, but small proteins like albumin and fluid can make it through. Every day, 20 liters of fluid, water, and protein seep out of the capillaries and becomes part of the interstitial fluid between the cells. About 17 liters gets quickly reabsorbed back into the capillaries, but that leaves around 3 liters of fluid behind in the tissues each day. This 3 liters of fluid needs to find a way back into the blood so that the body's interstitial fluid volume and blood volume both stay constant over time. That's where the lymphatic vessels, or just lymphatics, come into play. They collect excess interstitial fluid and return it to the blood. Once the interstitial fluid is in the lymphatic vessels, it's called lymph. You might be wondering how there can be 20 liters of fluid seeping out each day if the blood volume is only 5 liters. Well, remember that the 5 liters is constantly in motion, and that it gets recycled over and over in a single day. Unlike the circulatory system, the lymphatic system isn't a closed loop, because fluid and proteins make their way into the microscopic lymphatic capillaries, and all of the collected lymph is dumped into the veins. Lymphatic capillaries are the smallest lymphatic vessels, and they're located throughout the interstitial space. Lymphatic capillaries are extremely permeable because their walls are made of endothelial cells that only loosely overlap, forming one-way mini-valves. These endothelial cells are anchored to structures in the interstitial space by collagen filaments, which allows the lymphatic capillaries to stay flexible but retain their overall shape. When the pressure in the interstitial space is greater than the pressure in the lymphatic capillary, the endothelial mini-valves open up, allowing fluid to get in. When the pressure in the interstitial space is less than the pressure in the lymphatic capillary, the endothelial mini-valves are pushed shut, which keeps the lymph inside. Once that lymph is inside the lymphatic capillaries, it travels through bigger and thicker walled vessels, then trunks, and then ducts. There's no pump pushing lymph through the lymphatic system. Instead, smooth muscle in the lymph vessels reacts to the pulsing of nearby arteries by squeezing to get things started. And then the squeezing of skeletal muscles, which normally contract throughout the day, exert external pressure to keep the lymph flowing along, eventually reaching a nearby lymphatic trunk. To keep the lymph from sliding backwards, the lymphatic vessels have valves just like the veins do. The lymphatic trunks are each named after the regions of the body that they drain the lymph from. 2 lumbar trunks, 2 bronchomediastinal trunks, 2 subclavian trunks, and 2 jugular trunks, as well as 1 intestinal trunk. From there, the lymph is delivered to either the right lymphatic duct, which collects lymph from the right arm and the right side of the head and chest, or the thoracic duct, which is a lot bigger and collects lymph from the rest of the body. The right lymphatic duct dumps lymph into the junction of the right jugular vein and the right subclavian vein, and the thoracic duct dumps lymph into the same junction on the left side of the body. That particular spot is perfect because it's where the pressure is very low, which makes it easier for the lymph to flow in. The lymphatic system has key advantages. For example, it can pick up larger molecules, like hormones, that are too big to get into the capillaries, and then get them to the bloodstream. The lymphatic system can also help get nutrients to the tissues. For example, during a meal, fatty acids get packaged into balls of fat called chylomicrons by the small intestine. But just like the hormones, these are too big to move across capillaries. Instead, the chylomicrons go into special lymphatic vessels called lacteals, which get their name from the fact that the lymph that flows through them looks like milk. The chylomicrons slowly make their way up into the thoracic duct and get dumped into the venous blood. Now, the lymphatic system also plays a role in immune function. Lymphoid organs remove foreign material from the lymph to keep it from entering the bloodstream, and act as lookout points for the body's immune defenses. Some lymphoid organs are in the form of diffuse lymphoid tissue, where they're just a loose arrangement of lymphoid cells and protein, typically in the lining of the gastrointestinal and respiratory tract. Another type of lymphoid organs are lymph nodes, which are tightly packed balls of lymphoid cells and protein. Hundreds of lymph nodes cluster along the lymph vessels, each one a few millimeters to about 1 to 2 centimeters in size. When they're concentrated along the lymph trunks, you can feel them, especially in the neck, armpit, and groin. They can also be found in the intestinal wall, where they're called Peyer's patches. When an infection gets into the tissue, it can slip into a lymphatic capillary and move into a lymphatic vessel. Unfiltered lymph fluid drains into a nearby lymph node, where any pathogen or piece of a pathogen is quickly detected by a dendritic cell, which is a type of antigen-presenting cell that serves up pieces of anything it destroys to other immune cells. In the lymph nodes, dendritic cells continuously sample the lymph and present antigens that they come across to the B cells, which are a type of lymphocyte that can make antibodies. This is kind of like the transportation security looking at luggage at the airport, making sure that anything that goes through the lymph nodes is safe. B cells are designed to only react to foreign antigens, and if the dendritic cell presents something foreign, the B cell turns into a plasma cell and starts cranking out antibodies, which then flow into the lymph and exit the lymph node. There are also circulating T cells, which are another type of lymphocyte that move between the lymph nodes, lymph, and blood, on the lookout for pathogens and infected or abnormal cells that have been tagged by antibodies. Another lymphoid organ is the spleen, which is about the size of a fist and sits on the left side of the body below the diaphragm and on top of the stomach. The spleen has both white pulp and red pulp. The white pulp is where antibody-coated bacteria are filtered out of the circulation and antibodies are generated by B cells. In a sense, the white pulp of the spleen is like a giant lymph node, although unlike a lymph node which receives unfiltered lymphatic fluid, the spleen receives blood. The red pulp of the spleen is where old and defected blood cells are destroyed, and their parts, which are the hemoglobin, the heme chain, and the iron, are either broken down or recycled. The spleen is also helpful in that it keeps red blood cells and platelets available in case they're suddenly needed by the body. Really, it's an organ that's got your back in an emergency. Another lymphoid organ is the thymus, which is in the upper part of the chest, just below where a necklace might sit. The thymus is most active in the neonatal period and pre-adolescent years, and then slowly atrophies and gets replaced by fat after puberty. The thymus is involved in the development of T cells, making sure that any T cells that react to self-antigens, antigens that are normally found in the body, are promptly destroyed. A final set of lymphoid organs worth mentioning are the tonsils, which include the adenoid, tubal tonsils, palatine tonsils, and lingual tonsils. Together they form a ring of lymphoid tissue around the throat, and their main job is to trap pathogens from the food you eat and the air you inhale. Alright, as a quick recap, the lymphatic system refers to the one-way network of lymphatic vessels that allows lymph, which is a clear fluid that's squeezed out of the blood, to transport nutrients to cells and act as a method of waste removal. Lymph is cleansed at lymph nodes throughout the lymphatic system, which play an important role in immune function.
So those are kind of two more videos thrown in there to kind of help keep it a little more simple for us, because we're getting into pretty darn complicated topics, and there's a lot of T this and B this and a lot of acronyms and abbreviations floating around, which is a little more challenging. Okie dokie. We are kind of let's kind of back up together. We're now going into page 168 of your text, and we're starting to talk about the cells and tissues of the immune system in a little more detail. And they start by talking about B cells and how they develop in the bone marrow. So to recognize the vast array of different carbohydrates and proteins expressed by different pathogens with a high degree of specificity, an equally vast range of B cells, each with its own unique antigen specificity, must exist. So B cells recognize antigens through their BCR, B cell receptor. The BCR has the same structure and the same specificity as the antibody it goes on to secrete during an immune response. So we have billions of different B cells in our body, but each are able to respond to a unique antigen. For any lymphocyte, once its BCR or its TCR genes have been spliced together, they're committed to producing one antigen-recognizing protein, and that's it. The idea of kind of gene rearrangement and the generation of the BCR antibody. So how does the B cell receptor generate its antibody is what we're talking about here. So in order to produce such a wide variety of the BCRs, we now know that we have to be pretty tricky in the human body to circumvent the need for so many different specific types of antibodies. So the genes that encode the anti-binding region of the BCR are expressed in segments. So that's what we can see here. Dozens of different options for each gene segment combined together in a mix and match fashion, which is what they were referring to up here. And they create the great BCR diversity required with very few genes. So the process is called somatic gene rearrangement. This occurs in B cells, they're precursors in the bone marrow. So because the B cell receptor, the BCR, has two chains, a heavy and a light, each rearranges its genes, encoding the variable region independently. So that process increases the possibility of generating unique antigen binding sites on the receptor. Gene rearrangement does not depend on an antigen. So an antigen-specific B cell can develop well before an exposure to a given pathogen. So as the gene rearrangement process occurs, the new B cell receptors are tested. During development, the B cells with non-productive BCRs or self-reactive BCRs are deleted so that the B cells that survive are functional and unlikely to lead to autoimmune disease. In the really longest way possible, what they're trying to tell you is that our body is very smart, and we have figured out how to kind of make groupings of the genes. And then within that, there's a lot of different ways we can mix and match the grouping to create a different picture so that we can get the thousands of different cells that we need for all of the things we may encounter. To me, it makes me think of a Rubik's Cube. So each square on the Rubik's Cube is, if you lined it out and connected them end to end, it would be this super huge, long, really hard thing to manage. But instead, they're kind of packed into the little squares, and the little squares are like the Rubik's Cube. And then we now know how we can twist and turn and manipulate the Rubik's Cube. So we're still manipulating a whole lot of variabilities, but we have figured out a smart way in our genes to do that, to keep up with the demand of creating a really big, different amount of B cells. So I hope that is a little helpful to kind of understand what that means. And so what it's also trying to tell us is that as we are developing these different B cells, we are kind of auto-watching to make sure we create the right kinds of cells. And we know that that's very important, because if we were to be able to create faulty B cells, that's what can lead to things like autoimmune disease. So each day, billions of new B cells are entering the circulation from the bone marrow. The naive ones that are like colorless right now, right? They're not red, white, blue, yellow, or whatever that the colors of the Rubik's Cube are. They're clear, so to speak, and they will only survive for a few weeks if they do not encounter an antigen. So it's kind of like if they bump into the antigen, it goes from clear to an activated color specific to what antigen it encountered and in a specific pattern. And then our body knows to take all of those together and make the separate cubes of the Rubik's Cube, and then we can manipulate that now into a lot of different things. So what they're trying to tell us is we every day make billions of B cells as they come floating out. They're naive, they're blank or clear, is how I like to think of it. And then once they bump into an antigen, they pick up that color of the antigen and a specific pattern. And then they're used up by our smart genes to be put into different formulations, different chain configurations, to create a specific antigen that we need to be useful for. So if a clear, quote unquote, clear, naive B cell doesn't bump into something and pick up a color, so to speak, then it's going to die off. We do this process over and over again throughout our entire lifespan. Okay, so now let's talk a little bit about how we develop the T cells. So we talked about it, that in the video it noted that the thymus is most active in our early part of our life. And so the thymus is a primary lymphoid organ critical for T cell development in early life. So at birth, in a fully formed human, the thymus is fully developed. However, after 1 year of age, the thymus begins to involute and shrink, and its function and its usefulness begins to reduce. So if we have naive, inexperienced T cells that are generated by the thymus, these are long-lived and they are self-renewable, that live out in our periphery. This means that the loss of thymic function with age does not impair our T cell driven immunity over our lifetime. So another difference between a B cell and a T cell is that the B cells, we just mass produce them. Billions are thrown out there from our stem cells. They'll float around kind of quote unquote clear. That's my analogy. If they don't bump into an antigen and aren't useful, they get killed off. And we have to do this process over and over and over and over and over again. Whereas the T cell and the T cell receptors, when we make our T cells, these are done in the thymus, which is most active within our first year of life. And they are self-renewing, and they will live a really long time in the periphery. So as we naturally shrink down the thymus and we lose its function as we begin to age, it doesn't mean that we don't have active T cells, because these T cells live a really long time. Remember that if the progenitor cells from the bone marrow get to the thymus, they become thymocytes, cells that are committed to the T cell lineage. Okay? So back to this slide, if we come here and they become thymocytes, they're committed to this pathway. There's no going back to a different pathway. The generation of the TCR also have their own unique pathway to rearrange, and they rearrange their their chain segments here, XYZ. So they're very smart. It's very similar to the B cell part, that we figured out a way to pack them and combine them and rearrange them to meet the needs of the body. A T cell receptor is made up of two polypeptide chains, most commonly the TCR alpha chain and the TCR beta chain. Remember that the T cells are the hoity-toity ones, and they need something served up to them on a silver platter. They don't just go out there and randomly bump into antigens. They kind of wait for the antigen to be served to them. And those silver platters, remember, are the MHC molecules or the HLA complexes. Yes, each and every developing T cell will express multiple T cell receptors of a single specificity on its cell surface. So each T cell will recognize only one type of antigenic peptide, and most T cells will express the alpha beta TCR, which is made up of the alpha and beta chains. But we do have a small number of T cells that will express the different type of receptors made up of the different chains. And those are a little bit different. Developing T cells commit to becoming then either a CD4 cell, which is the helper cell, or a CD8 cell in the thymus. And they do that by expressing either the CD4 or the CD8 co-receptor along with the T cell receptor, and they come together and they commit to either be a CD4 or a CD8. Although these co-receptors do not bind directly on the antigen, they do bind to the MHC complex that present the antigen to the TCR. And because a CD4 T cell only recognizes MHC class 2 and CD8 cells only recognize MHC class 1, the type of peptide antigens recognized then by the CD4 T cell and the CD8 T cell are different. Once these are matured, the CD4 and the CD8 cells go on to play very distinct roles in our immune response, which we've already kind of figured out. Okay. I know this is a lot of information and confusing, but we're really trying to understand the differences between the B cells and the T cells. We watched that video about the lymph circulation, which I think is always very helpful. So the lymph system is very important, obviously, in our immune system, because this is often the site of where the B cell and the T cell will encounter things. So the B cells and the T cells often encounter the antigen in either the blood or the tissues. They will travel to lymph nodes, spleen, and our other secondary lymphoid tissues. They will interact in follicles surrounded by the paracortical zone. The T follicular helper cells, which we learned about in that video, promote activation of the B cells, enhancing differentiation and antibody production. And then remember, we learned about that we have some of those gut-associated lymph nodes and spleen-associated lymph nodes, and we have these kind of interactions that are going to produce us a very specific type of antibody production. I'll add this to this slide up here. I would recommend printing off the transcript of these videos. They are available to you as transcript written format to have for reference. I think that would be really good, just like you're going to have your notes or your slides here, being able to reference back to the transcript of the osmosis videos could be very helpful. I really appreciate how osmosis talked about the spleen and gave a very quick rundown of that and the differences of the pulp, the red pulp, the white pulp, etc. So remember that in the red pulp of the spleen, that is where damaged or senescent red blood cells are removed. And senescent ones mean they're programmed for death. That's where the elderly go to their nursing home to die. Or if they're damaged, they'll go there as well. And then within the white pulp nodules is where the T and B cells are going to respond to the blood-borne pathogen. And that would be like figure 6.9 in your book. Like lymph nodes, the white pulp of our spleen is organized into clear T and B cell areas. So once the T cells and the B cells enter into the white pulp, they definitely go left or they go right and they stick to their areas. The T cells are mainly located near the arterial and the periarteriolar lymphoid sheath, and the B cells like to live within the lymphoid follicle, which is adjacent to the T cells. So when you hear about the marginal zone of the spleen, that's a very unique feature. The marginal zone surrounds the white pulp and is bordered by the perifollicular zone. Resident within the marginal zone are specialized macrophages as well as innate-like B cells known as our marginal zone B cells, which are clonally distinct from our typical adaptive B cells. In humans, the marginal zone B cells can circulate throughout the body, and they are associated with protection from bacteria and viral infections, including HIV. So that's why I bring this up. As we talk about HIV, or if you work with HIV, you will hear about the marginal zone B cells and the differences there. So I did want you to hear that at least one point in your life of where that to come from. Remember, the thing about the spleen is that once it is injured or ruptures and actually bursts, the spleen may be removed in an emergency. But if you have your spleen removed or you're born without one, those patients are very much at a risk of developing severe bacterial infections because we removed this whole area where we store a lot of our immune system cells. Not a good thing. Also, to throw out there again, the video did a nice job of talking about it, but that we do have gut-associated lymphoid tissue, which is referred to as GALT. So the gut-associated lymphoid tissue compromises 3 secondary lymphoid type tissues. Remember, the Peyer's patches, the isolated lymphoid follicles, and the mesenteric lymph nodes. So those gut-associated lymphoid tissues is the largest lymphoid organ in the human body. Its function is to support tolerance to a highly diverse commensal gut microorganism. Okay, so if we wanted to say, why are Peyer's patches important? If you were asked a question like that, you would say, because that is part of what compromises the gut-associated lymphoid tissue. And those are really important because that's actually our largest organ in the body. And it is what we need to support the tolerance to the highly diverse commensal gut microorganisms, so the good gut bacteria, but also while maintaining the ability to respond to foodborne pathogens. So it's important to remember that even within our GI tract, we have a very specific type of immunity and an immune system that works very closely with our lymph node system. Outside of the secondary lymphoid tissues, effector lymphocytes are scattered throughout the gut epithelium and the lamina propria. In addition to their presence in the blood and the lymphoid tissues, the cells of the immune system otherwise are found throughout the body. Both innate and adaptive immune cells are strategically positioned within the peripheral tissues, within the fat and organs, as well as within our skin and our mucosa. And their sole purpose then is to combat the invading pathogens, neutralize toxins, and kill cancer cells. So we're reviewing again. We've reviewed a bunch about the barriers of the skin and the mucous membranes. We now talked about the differences between innate immunity and adaptive immunity. To summarize the principles of innate immunity, this is a really nice slide. The innate immunity works by releasing internal signals of cell injury and death, and that the pathogen invasion via trauma or environmental exposure brings pathogen-associated molecular pathways. So I want to jump back to that previous video we were on and kind of finish to bring this together and tie this up. So we're going to jump back in here. This is the innate immune system video, and kind of listen to it again with this perspective now, having talked a little bit more about these things.
Since macrophages live in the tissue, they start recognizing pathogens within minutes of an infection. And the way that a macrophage figures out if something is a healthy host cell or a pathogen is by the molecules that a cell or pathogen has on its surface. This is because cells of the innate immune response don't distinguish one invader from another invader. This probably won't come as a surprise, but pathogens have molecules that humans don't have, and they're called pathogen-associated molecular patterns, or PAMPs. PAMPs include bacterial wall components like peptidoglycan, lipopolysaccharide, or LPS, and lipoteichoic acid. PAMPs also include fungal wall components like mannan and flagella proteins, which can be found on some parasites and bacteria. For intracellular pathogens like viruses, PAMPs might include the viral RNA or DNA. Now, PAMPs are recognized by pattern recognition receptors, or PRRs, which are receptors on various immune cells including macrophages, neutrophils, eosinophils, basophils, and mast cells. There are two main groups of PRRs, phagocytic PRRs and signaling PRRs. Phagocytic PRRs bind to PAMPs so that a phagocyte can gobble it up, but they do not allow for cytokines to be released to other cells. And this is a super important point, because it allows the macrophage to investigate and eliminate the threat before signaling that there's an invader. This is kind of like investigating a noise before calling the cops. After all, it might just be a chicken wandering around on the roof. So if there are just a few pathogens around, then the phagocytic PRRs will get activated, and phagocytes will eliminate the pathogens. Going back to our garbage truck macrophage, let's say that its PRR recognizes a PAMP on a bacterial cell. It'll then swallow up the bacteria and trap it in a vesicle called a phagosome. The phagosome then fuses with another vesicle called the lysosome and forms a phagolysosome. At this point, the phagocyte will undergo a variety of chemical reactions to kill the pathogen. First off, the phagolysosome contains two types of granules, specific granules and aerophilic granules, which help destroy the bacteria. The specific granules go to work first, and they have proteases and hydrolases which are active at a neutral pH. As the organisms die, potassium and hydrogen ions are drawn into the phagolysosome, which decreases the pH, and this allows the enzymes in the aerophilic granules to go to work. Overall, the enzymes in these granules kill about 2% of the pathogens that the macrophage eats. The other 98% of the pathogens that the macrophage ingests get killed by a process known as oxidative or respiratory burst, which is a process that's so powerful that it eventually destroys the phagolysosome and the macrophage as well. Phagocytes, like macrophages and neutrophils, are capable of creating an oxidative burst, but neutrophils are really good at it. An oxidative burst leads to the production of nitric oxide, or NO, and reactive oxygen species, which are chemically unstable molecules that contain oxygen, like superoxide anion and hydrogen peroxide. These molecules can bind to molecules within the cell, and that can lead to tissue damage. The oxidative burst gets underway when the phagolysosome forms, because that activates the enzyme NADPH oxidase. NADPH oxidase transfers an electron to two oxygen molecules, creating NADP+, two superoxide anions, and a hydrogen ion. The enzyme superoxide dismutase then converts the hydrogen ion and superoxide anions into hydrogen peroxide, which is highly toxic to microorganisms. Also, in the presence of NADPH and oxygen, arginine is converted into nitric oxide by an enzyme called inducible nitric oxide synthase, or INOS. Nitric oxide and reactive oxygen species destroy pathogens, but also destroy the phagocyte as well. And as it turns out, being deficient in NADPH oxidase allows bacteria and fungi to escape getting killed by neutrophils, and this can lead to chronic granulomatous disease, or CGD. Now, let's say that there are a lot of pathogens around. In that situation, the tissue-resident phagocytes will need some help to deal with this larger invasion. This is where signaling PRRs come in. Both phagocytic PRRs and signaling PRRs will be activated. And if the amount of pathogens is high, the signaling PRRs will signal to the body to send reinforcements to the location. Signaling PRRs don't help the macrophage to gobble up a pathogen, but they do get the macrophage to release cytokines, kind of like lighting a signal fire to start the immune response. Cytokines are small secreted proteins that cells use to communicate with each other. The most important group of signaling PRRs are the toll-like receptors, called TLRs. TLRs are found on all leukocytes, as well as on epithelial and endothelial cells. TLRs 1, 2, 4, 5, and 6 are located on the cell surface, and they bind to PAMPs like peptidoglycan, lipopolysaccharide, and flagella. TLRs 3, 7, 8, and 9 are found within the cell, and they bind to intracellular PAMPs, like viral RNA. Now, when any of these TLRs gets activated by a PAMP, they activate the transcription factor nuclear factor kappa light chain enhancer of activated B cells, or NF-kappa B, which stimulates the secretion of pro-inflammatory cytokines like tumor necrosis factor alpha, interleukin-1 beta, and interleukin-6. These cytokines cause vasodilation, bring more leukocytes to the area, and induce fever. If the pathogen is intracellular, the cell starts to secrete interferon alpha and interferon beta, which interfere with the virus's ability to replicate in nearby cells. Alright, as a quick recap. Our cells recognize pathogens by using pattern recognition receptors, or PRRs, which identify pathogen-associated microbial patterns, or PAMPs, on the pathogen. Phagocytic PRRs help phagocytes like macrophages and neutrophils ingest pathogens, which they destroy using specific endoxerophilic granules or the respiratory burst. Signaling PRRs, like TLRs, help cells to bind to PAMPs on the surface of a pathogen, or bind to intracellular pathogens, and stimulate cytokine production, which brings more immune cells to the site of inflammation.
So I want to point out here that leads us right into how our immunity system ties into then wound healing XYZ, and that these processes are all very specifically and well timed, so that once we have these mast cells and the macrophages are doing their jobs, then the blood vessels dilate and permeability increases and the white cells are allowed to come to the area again, and then you have these cardinal signs of acute inflammation will develop, and then you will end up with tissue repair and wound healing. So these things are all really important to have happen so that we can get inflammation and bring the next steps together, so to speak. So that leads us into the idea of inflammation, and again, understanding the process of how inflammation works in our body. So acute inflammation is a consequence of our innate immune system, okay? Because of the way the immune system works, that is the next step it's going to do is trigger into this next step of the the acute inflammatory response. So when you think inflammation, we think mast cells and macrophages. We also have something called sentinel cells that are there to help detect tissue damage and invasion. And when those all are working together, so once acute inflammation happens, then that will trigger the inflammatory cascade. So we have to kind of figure this out of how this all works. The detection of danger by the cellular pattern recognition receptors, those PRRs, which is what they talked about in the video, those are necessary, and as well as our mast cells and our macrophages. All of these things need to happen together to trigger the components of the inflammatory cascade and then triggering the components of the complement system. And all of that leads to the production of chemotactic signals that stimulate the movement of leukocytes, mainly our neutrophils and our monocytes, from the circulation into the tissue at the site of injury. Activated mast cells and activated macrophages at the site of injury or infection release chemical mediators like histamine that will increase the blood vessel permeability and cause local blood vessels to dilate. The area is going to have an increase in the blood flow, and so then we get that redness and erythema. The endothelial cells lining the blood vessels upregulate membrane proteins that bind to leukocytes to assist their movement from blood to tissue. What does that mean? So then at the endothelial cells that line the blood vessels, which is kind of what we have here, they are going to do something different to upregulate the membrane proteins, right? And then those are going to bind to the leukocytes, and they're actually going to kind of like hold their hand and drag them in to move them from the blood to the tissue where they're needed. Because once in the tissue, neutrophils and monocytes differentiate into a highly active state, and that means that they have increased ability to phagocytize the pathogens and generate additional mediators of an inflammation. Activated phagocytes kill the microbe and contribute to the escalating vasodilation, which then allows for leukocyte recruitment and inflammation to come in. And this is the process then that's all happening. It's a lot of things happening. So this one in your book is under figure 6.10. This is the sequence of inflammation. So this is A, and this is describing the detection of danger. So this has happened. The wall has been damaged. This wall has opened up with the epithelial and allowed all of these things. Our PRRs have been pattern recognized receptors in the tissues have been activated. The macrophages and the mast cell and all of these things are now being triggered. When we get to the next step, which is the leukocyte recruitment and elimination of the stimuli, which is what we see here, you also notice that we're beginning to form a fibrin band here from the area of injury. Now we can see in here we've got some complement factors going on. Our monocyte or neutrophil, we've got some antibodies in here. We've already starting the phagocytosis of the pathogen and cellular debris. Our neutrophils are now coming in, which leads us to then C, which is resolution. So now again, we have this plug here. It's much less busy. Things are happening. Phagocytes, now the apoptic neutrophils, the neutrophils that have done its job, are going to be able to die off and get removed. And then that leads us then to wound healing. And at this step, the macrophages secrete the fibroblast growth factor and the vascular endothelial growth factor. And so now we've got, again, re-epithelialization, that the cell wall is closed, the tissues and the capillaries are replaced, and now we have that complete wound healed. So we start by specifically talking about each of these different immune cells of the innate system, right? So we've talked about neutrophils, macrophages, monocytes, etc. So now we're going to talk about these in more detail of each different type of cell. So we start with the neutrophils. It's really confusing because they have so many different names. Why does the neutrophils also have to have polymorphonuclear leukocytes or PNLs? There's just a lot of different names for things. It really is making me crazy sometimes in practice. The neutrophils, the most abundant white blood cell. So when you say white blood cell count, what we're talking about is neutrophils, leukocytes, monocytes, eosinophils, etc., and monocytes. So the total white blood cell is a combination of all the different types of white blood cells. So in this case, we're talking about neutrophils. So of the white blood cells, the neutrophil is the most abundant. The numbers will increase, obviously, in acute infection and inflammation. And that's when we see what we call leukocytosis. So again, the neutrophils and the leukocytes are what we're talking about here. And when you see a sharp increase in the leukocytes, that's why we call it leukocytosis. That should signal us as practitioners to begin to think of infection or inflammation that must be happening to drive that white blood cell count up. The lifespan of the neutrophil is about 24 hours. They spend about 12 hours in circulation, and they spend about 12 hours in the tissue. And then once they're used and done, they're scheduled to be killed off and will be removed. The neutrophil contain the granules with these protease enzymes and other granules that will synthesize reactive oxygen species, which is what the video talked about, to kill the bacteria. The mobile, highly active phagocytes are able to attach to blood vessel walls and leave for the tissues by diapedesis, which is another reaction following the chemotactic signals. So these are some really nice images here to help describe the neutrophils. Okay. Neutrophils are highly phagocytic cells, and they are able to ingest and kill thousands of bacteria before dying, so they're highly useful. They're pretty darn effective. They have two mechanisms of bactericidal activity, so they actually kill the bacteria. They can do that with exposure of the bacteria to toxic oxygen compounds known as those reactive oxygen species. And they can also have degradation of the bacteria by the protease and these other enzymes. The specialized neutrophil organelles and enzymes generate the reactive oxygen species as oxygen-derived free radicals burst and help this process. So together with the reactive oxygen species, the ROS, the phagocyte lysosomes contain many proteases that are able to break down the bacterial proteins, and we're ultimately killing the bacteria. So these chemicals are very efficient in eliminating the pathogen. However, they're also highly toxic and can potentially cause secondary tissue damage to the host, like inflammation. Consequently, the oxidative burst is limited to states of strong neutrophil activation, resulting from the presence of bacteria. So we kind of have that protective mechanism built in, that we're not just going to have this super toxic chemical floating around as neutrophils. The toxicity kind of comes only when they're highly gathered in high numbers, which happens in response to a bacteria. The body has endogenous processes protecting against this process, right? So that's what we're talking about here. One of the examples of these compounds that can help protect from this bad part of the neutrophils is the enzyme alpha-1 antitrypsin, which counteracts the action of some of these particular proteases. Kind of newer, quote unquote, in science is that we've actually discovered an additional defense mechanism restricted to neutrophils, so specific to the neutrophils. When faced with overwhelming numbers of bacteria, neutrophils can combine and organize themselves into neutrophil extracellular traps, which is what we see here. Activated neutrophils in the process of dying, so they've done their job, they've gobbled up a ton of bacteria's material and proteins, so they've done their job, they're now in the process of dying, and when those release their DNA into the extracellular space, it generates a sticky trap for bacteria, which is what we can see here, okay? So the neutrophil's done its job, it's beginning to break down, and it's scheduled to die off. It will create this neutrophil extracellular trap. Okay. Antimicrobial enzymes and the ROS contained in the neutrophil granules are also released into the extracellular compartment, and that targets the entrapped bacteria for destruction. So long story short, we now have figured out there's this extra part that happens during the neutrophil cell death that form this sticky area that works as a neutrophil extracellular trap, and it's going to help trap essentially more bacteria. These NETs are implicated in the pathophysiology of cystic fibrosis complications and some of our autoimmune diseases. Okay, so even though our neutrophils are short-lived, they can undergo extensive transcriptional remodeling as they move from the bone marrow into the periphery, so they have a whole bunch of variability in them as well. Neutrophils, are going to be, as we're talking about, they're activated by the LPS, the PAMPs, or the toll-like receptors, which was very well done in our video. And the neutrophil activation is promoted by the inflammatory mediators. So these are all tying together now. between our immune response. Our innate immune response has to work closely with the inflammation process to get the job done, so to speak, and help bring the neutrophils to the area. This is a pretty cool thing that shows the neutrophil extracellular trap. Which is really cool because it's filled then with these bactericidal molecules that are actually going to be toxic to the bacteria and shrivel them up and cause them to die. Okay, so we've wrapped up neutrophils. That was fast and heavy neutrophils. Now we're going to move on to basophils, mast cells, and eosinophils. These are non-neutrophil granulocytes. They're non-phagocytic, and they degranulate upon stimulation, and the effects of them are dictated by their granule contents. So the differences between them, even though they act very much the same, is they're each filled with different granule content. The mast cell and the basophil granules contain histamine. So anytime we're talking about in the future an antihistamine, an allergy, anaphylaxis reaction, when we want to use antihistamines, what we're talking about is that we have this process where the mast cell and the basophil have probably been activated. So they both contain histamine, and we know that histamine is important in the inflammation-induced vasodilation and causes that increased capillary permeability. Eosinophils are the least numerous, and they're really only there specialized to kill parasites. So if you have a high number of eosinophils, something is very wonky and not going well, there's a cause for that. Sometimes it's medication, there's a whole bunch of different things, but you need to pay attention if you ever have a whole bunch of eosinophils because something's gone wrong. An allergy, something has gone off. So it could be from a parasite. So attached to parasite and exocytosis granules with killing proteins. So that is a possibility of what can cause your eosinophils to grow in numbers. What we see most typically is people with allergies. So vulnerable tissues usually have eosinophils present, so not present in normal tissues and those without allergies. Basophils are considered somewhat rare, but they do circulate in the bloodstream. But mast cells reside in tissues. They're all over the place. They're very active or found in connective tissue of the skin. We have them on our mucosal surfaces, and they tend to live kind of adjacent to blood vessels. The basophils and the mast cells, again, contain those granules of pro-inflammatory chemicals such as the histamine. Immediately after tissue injury or during the early phase of an allergic reaction, the basophils and the mast cells will degranulate and they'll release that histamine, and histamine's a potent vasodilator. So if you're having an allergic response, if we use antihistamines quickly, that can really counteract the allergy response. Eosinophils are a bit more like neutrophils, so they are phagocytic granulocytes. Eosinophil cytoplasm is filled with the granules containing the toxic enzymes that are released by exocytosis onto specific targets. Eosinophil granules are capable of killing larger targets, such as think parasites, which are too big for phagocytosis. But eosinophils have a critical role in allergy and hypersensitivity reactions because they tend to participate in the late phases of allergic responses. So, for example, in asthma, eosinophils in the lung is what contributes to airway hyperresponsiveness, then extra mucus production, tissue damage, and then airway remodeling. Not a good thing. Next group of cells are the monocytes and macrophages. The monocytes, macrophages, and dendritic cells represent the group of the highly phagocytic cells amongst all of these. Monocytes generated in the bone marrow will enter the bloodstream where they circulate as an immature form of a macrophage. The monocytes migrate into tissues before they differentiate into macrophages. In tissue, when they're in there, the monocytes increase their size and phagocytic capacity. So we have two types of macrophages. The first type operates beneath the epithelial tissue and also in several organs, and they are patrolling as these guards, ready to ingest any microorganism able to get through the tissue. So they're kind of walking the perimeter of the fence, and they're ready to eat it up if they get through that fence. The second type of the macrophage circulates between the lymph and the bloodstream. They live in that area, and they're also conducting surveillance for any foreign material. Some cells of the macrophage lineage reside in specific tissues and specific tissue locations to exert their function, and they have very unique names, such as if they're in the lung, they're called an alveolar macrophage. If they're in the bone, there are osteoclasts, etc. These are all listed in table 6.3 in your macrophages. As an example found in the liver, are known as Kupffer cells. Macrophages are also located throughout the primary and secondary lymphoid tissues, which is what we've talked about. And there they exhibit a range of different phenotypes and functions, and that's depending on their specific location. Similar to macrophages found throughout the lymphoid tissues, some of them function and exist within the specialized tissue resident macrophage lineages. A whole lot of words. Remember, when an infection or an injury occurs, these soldier out there sentinel macrophages detect cell damage and migrate to that area by a process called chemotaxis. Chemotaxis is the movement of a cell towards or away from a chemical stimulus. Phagocytes use pseudopods to move toward a microorganism, as an example. And the pseudopods are temporary cytoplasmic extensions that reach out from the cell to allow movement. Okay. Chemostatic substances that attract the monocytes and the macrophages include the microbes' products, components of damaged cells, chemicals released by the other white blood cells, and the peptides that are derived from the complement system will attract the monocytes and the macrophages. When they're activated, the pro-inflammatory macrophages begin to secrete cytoxins, in particular interleukin-1 and the tumor necrosis factor. And that contributes to chemotaxis and activates other leukocytes and helps bring them to the area. It also serves as a signal to the bone marrow to produce more leukocytes. And then as the infection or the injury is beginning to resolve, anti-inflammatory macrophages will support the wound healing by actually removing the old debris and then also helping stimulate tissue repair. Also to remember is that activated macrophages are the major source of the inflammatory cytokines, like we talked about, some of which require activation of an intracellular inflammasome for secretion. So that is a weird word you may hear. In addition to activating other cells, some of these cytokines are what actually help cause that systemic fever. That's what triggers the malaise and the anorexia. So by activating some of these other cells, the monocytes and macrophages help signal externally that our body's having trouble. So it gives us these feelings of fever and fatigue and malaise. Also to remember, the dendritic cell is the most critical cell type for communication between the innate and adaptive cells, arms of our immune system. So say that again. The dendritic cells, remember I said, could come from both lineages, and they also can be what communicates. They're the line of communication between our innate immune system and our adaptive immune system. So the dendritic cells are critically important that these two systems can work together. Remember again, like I said, they can come from either the myeloid or the lymphoid precursors, and they're located throughout the body. These are also in the skin, which is where we get the Langerhans cells and the dermal dendritic cells. They can be in primary lymphoid tissue and secondary lymphoid tissue, and as well as within the blood. Immature dendritic cells are highly phagocytotic. Sorry, go back up in here. I do want to point out the book also talks about opsonization and phagocytosis. And I don't know why they didn't have a slide in here. So take some notes on this. The neutrophils, macrophages, and the dendritic cells are able to conduct phagocytosis of pathogens as one of their functions. If you were to be asked what is the functions of phagocytosis, what's the process? Refer to figure 6.13. This literally walks you through step by step. The ability for phagocytosis to happen depends on the ability of the professional phagocytes, like the macrophages, to deform their plasma membrane to move towards the microbe by that pseudopod formation. Then they need to attach to the microbe, which is the process called opsonization. So the microbes have to get there, and then they have to be able to attach to the microbe through opsonization. Then they have to ingest the microbe, fuse the phagosome with an intracellular lysosome, and then they end up killing off the microbe, breaking it down into smaller tissues. That's why the formation of a pseudopod can be important to help bring them to the area and get them to the place they need to be. All right. Next, they kind of jump into this slide here, and we have talked a lot about this already, and the differences between the innate and the adaptive and how they talk together. We've reviewed the phagocytosis and how that works, and then that the dendritic cells will circulate to help be available for use as well. Hang in there. So the innate lymphoid cells, the ILCs, these were also talked about in our previous videos. If you're feeling really lost right now and like, holy crap, Sarah's talking forever and there's a ton of material, stop listening and go back and listen to the lectures again. Those 2 videos by osmosis are great. And listen to it or take notes off of it again, print out the transcripts if you need to, and reread the material. And so I do encourage you to take a break if you need to, and then come back to hear this next part. So the ILCs are a group of the innate cells that derive from that lymphoid lineage, and like the CD4 T helper cells, their main function is to produce cytokines. But what's different than the T cells is that the innate lymphoid cells, the ILCs, do not express a genetically recombined antigen receptor, and they don't express the lineage markers of other immune cells. What they do is they respond to their local environments by recognizing cytokines and chemokines and other signals through receptors on their cell surface. This is important because they function to augment the non-specific reactions of innate immunity. Remember, we're talking about B cells and T cells are highly specific and highly hoity-toity, and they really are only going to do what they want to do. The innate lymphoid cells are helpful in their way because they aren't hoity-toity, and they can augment the non-specific reactions of the innate immune system, so they help broaden that innate immune system better. The innate lymphoid cells, the ILCs, are categorized into 3 types based upon their function and their cytokine expression. So we have type 1 ILCs, which they also went through in the video. These include the helper and cytotoxic cells that produce the cytokine interferon gamma. The type 2 ILCs are found in fat, liver, lung, and skin, and they're important for fighting helminth or parasitic infection, but they also may mediate asthma and wound repair. And then the type 3 ILCs are found in the gut and the lung. And remember, these are important for maintaining homeostasis because we have to keep that natural balance between good and bad bacteria anyway. The ILCs also include the lymphoid tissue inducer cells, which are critical for establishing secondary lymphoid structures during development, and cytotoxic natural killer cells. Natural killer cells were the first lymphocytes shown to act more like innate immune cells. They were previously called a different name. They have killing granules, and they can attack both virus-infected cells and cancerous cells. We know more about the natural killer cells because we've learned about them a long time ago. They can recognize and directly kill cells that appear normal, such as a virus or an infected tumor cell. Armed by a series of surface receptors and cytoplasmic granules containing toxic substances, so they're really well armed, natural killer cells scan for the presence of unusual ligands on cell plasma membranes, which we talked about in the video. The ligand and receptor interactions then will modulate NK cell effector properties. The example the book gives is that the absence of the normal MHC complex class 1 expression on the surface of a target cell is not a good thing, which can occur in viruses or certain tumors. And that can induce the activation, though, of the natural killer cells. Once they're activated, the natural killer cells orientate and release their granules in the extracellular space towards the target cells. The granules contain mainly two substances, which is perforin and granzyme B. Perforin creates a channel in the plasma membrane, altering the membrane integrity and inducing cytolysis of the target cell. Membrane pores allow the entrance of the second compound, which is the granzyme B, which is a protease able to engage the apoptosis path leading to the cell destruction. So they kill. They're killer cells. And this cell helps us understand that we have ILC2 and where they're found and what they do, and then ILC3 where they're found and what they do. So this goes into the question, how are pathogens detected by our innate immune cells? The pathogen feature that differs from human cells. So how do we know what we're supposed to attack, right? These are recognized by TLRs, which is a class of the PRRs. So TLRs are subset pattern recognition receptors that mediate pathogen recognition in the innate immune system. There's many types and combinations of the TLRs. This is what this is representing here in this illustration. Many TLRs will recognize components of bacteria, yeast, or parasite cell membranes, while other TLRs are found on endosomes, and they recognize components of invading bacteria and viruses. This is a non-selective way of distinguishing between self and non-self. Remember, that is the main characteristics of the innate immune system. The cells of the innate immunity can also recognize and become activated by signals of tissue damage, cellular stress, and cellular death. So when we're talking about triggering the innate immune system, these triggers can occur in the presence or absence of infection. So that's a little scary, right? And they are referred to collectively as the danger-associated molecular pathway, the DAMPs. So when tissue damage occurs without a pathogen, without an invasive thing, so there's no infection, there's no pathogen noted. The book says, think of a bad ankle sprain that occurs without a break in the skin. The innate immune system response is referred to as sterile inflammation. And the DAMPs are the originating signals. The DAMPs are also recognized by the PRRs and can trigger the formation of the intracellular multiprotein complexes. So those multiprotein complexes are a lot. These are a lot of big words and a lot of things to talk about. But I did want you to understand this basic part here that the innate immune system has a pattern of recognition to understand what's a pathogen, but also sometimes when things can happen and be triggered even when there's not a pathogen present. The book does talk a lot more about the inflammatory pathway that, again, did not really make it onto the slide, but I do want to make sure you guys are reviewing this because it talks about some important things. The cardinal signs of inflammation, right, are redness, swelling, and heat. Sometimes it's also loss of function. So go through the sequence of inflammation and inflammatory mediators in your reading as well. This is where we get the idea that we have acute phase reactive proteins like the CRP, and so that these are non-specific things that tell us that something's going on. The CRP is going to rise when things like histamines and prostaglandins and kinines and leukotrienes are present. It's going to say, oh, something's not right. And the general reaction, it's a non-specific acute phase reactant, would be the CRP beginning to go up. So last part of this first part of the last part of part 1 is discussing the complement system. This is a series of proteins that are produced by the liver and can be found throughout the body in extracellular fluids, in our lymph system, lymph fluids, and in our blood. Many of the complement proteins are proteolytic enzymes that exist in an inactive state. They're similar to the proteins like we find in the coagulation cascade. So when it's activated by signals of pathogen invasion, the complement proteins initiate a cascade of activation whereby one enzyme cleaves and activates the next in the series. So it's literally the domino effect, okay? There's 3 different pathways that are able to activate complement, and that's really kind of the thing to understand about complement is what pathway you're on. So you'll hear about this, the different pathways. When you go to box 6.4 in your book, we talk about like page 182. The box 6.4 is the overview of the complement pathway, and I think that's important to talk about and to read about. So complement proteins are designated with an uppercase C and then are numbered 1 through 9 based on the order of discovery, not on the order in which they act, which is super annoying. Complement proteins that are activated by cleavage, meaning they cut in half and they'll produce 2 subunits, are indicated with the lowercase letters of A or B. There are 3 different pathways, and so that's in figure 6.17. which we have here. So the pathways are noted here. In the classical pathway over here, the bacteria are coded by antibodies. So they're signaled, they're labeled, and that attracts and activates the complement cascade. So very straightforward. It's antibody dependent. They attract C1, 4, and 2. And then they also have these fragments that split Z. Okay, great. Sounds good. The next pathway is the alternative pathway, which is over here. In the alternative pathway, complement binds to the bacterial cell wall, initiating its own cascade. So it doesn't wait for a coded antibody to tell us where to go. It goes and finds the bacteria itself, and then the cascade begins. The lectin pathway depends on the MBL, which is that circulating protein synthesized by the liver, and it waits for the MBL to attach to the bacteria before activating the complement system. Great. So either it's the classical pathway and you get to the C3 convertase, or the MBL triggers this pathway, which goes to the C3 convertase, or the complement system itself goes and tracks down the bacteria, and that triggers the C3 convertase. And then you get to this central part here. So then after activation, no matter what way you get there, then we get this process here. So C3 cleavage releases to C3A and C3B. C3B then cleaves down to C5. The free-floating C3A comes over here to form anaphylatoxins over here. The C5 going down the pathway cleaves to release C5A, which also leads to that. And then C5B continues down here, and then C5B is joined with these others over here, and together they form the C5B-9 MAC, and that leads to the cell lysis. I'm going to stop here. So we're ending at this spot here, ending with discussion of complement. There's only a few slides left, but this has been a lot of material and I want to break here. When you come back for the next session, we'll finish out these slides and start the second part of this chapter to finish it out. Thanks for hanging in there.